Hello, everyone, and welcome to today's webinar titled Patterns of Intrahost and Interhost Variation in SARS-CoV-2. This webinar is part of the ongoing coronavirus webinar series. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. So let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of your presentation window and follow the process to obtain your credits. Without further ado, I now present today's speaker, Dr. Todd Treangen, Assistant Professor, Computer Science at Rice University and the co-lead COVID-19 International Research Team. For a complete biography of our speaker, you can go to the Biography tab at the top of your screen. Dr. Treangen, you may now begin your presentation. Welcome, sir.
Great. Thanks so much. And thanks for this opportunity to present today. So I'll just get right into it. And that was a great introduction. Thank you, Susie. Thanks to everyone at Labroots for making this happen. So today, as was mentioned, I will be discussing patterns of within person, intrahost, another way of saying that, variation, and SARS-CoV-2. And as was mentioned, I'm an assistant professor in the computer science department at Rice University in Houston, Texas. So just get right into it. So just to get started, here's a high-level outline I wanted to share with you before we get into it. And basically, this is divided into 6 parts. And I'll start off with the introduction background on kind of motivation and concepts that I'll discuss. I'll then get into a high-level summary of what I mean by interhost variant landscape. I will present that. I will do some comparative analyses of some of these terms that I'll discuss throughout the presentation, and then talk about some of the potential impact of what these SNPs or SNPs and ISNVs are on molecular testing and utility of these for potentially tracking transmission of COVID-19. And then I'll offer some concluding remarks. So let's just go ahead and get started. So just as was mentioned, I wanted to reemphasize the learning objectives of this talk, as it kind of 3 main things I wanted to highlight. By the end, those listening to this presentation should be able to think about differentiating consensus level versus within host variation of SARS-CoV-2, what that means, how we can go about doing that. As a learning objective number 2, I would like everyone to think about and learn what's the potential impact of these single nucleotide variations on COVID-19 diagnostics. And as a third and final learning objective is kind of to understand the utility of these minor variants or intrahost single nucleotide variants on tracking SARS-CoV-2 transmission. So those are the 3 primary objectives. And then, so we'll get right into it. So just part 1, background. So historical perspective, I thought this was a very interesting and useful way to start it off is back in 2003, December, there was a paper published in Nature Reviews Microbiology by the co-authors listed on the screen titled SARS, Beginning to Understand a New Virus. And so here we sit, 17 and, years later, more or less, and I feel like you could replace that with SARS-CoV-2, beginning to understand a new virus. And so I'll start to get into what I mean by that, but I thought this was an appropriate way to begin this. This is related and kind of other related to previous efforts and going to take a deep dive into SARS-CoV-2. So just wanted to highlight this. So background, I'm not going to get into all of the biology behind SARS-CoV-2 and COVID-19, but I will highlight a few things, very brief. On March 11th, 2020, the WHO determined that an outbreak of a novel coronavirus, now that we know is called SARS-CoV-2, it began in Wuhan, China. late December 2019, and it had reached pandemic status. Initial analyses of these few initial genomes that became available indicated a few things, but then it was mutational rate was similar to other coronaviruses. So as data was arriving, we were thinking that this looked similar to other coronaviruses, but it was still kind of, as you get data, we were interpreting it. But one important point is important in order to properly assess the diversity of any RNA virus, and specifically SARS-CoV-2. It's really important and necessary to consider the intrahost variation as well. And that's kind of one of the main points of this whole talk that I'm giving today. So I just wanted to highlight that. And that's and segueing in from that, we're high level. And again, there's not going to be tons of biology here, but one slide will dedicate to this. This is looks like this is a figure from Stadler et al. And this is describing high level what the SARS-CoV-2, or it's actually the SARS-CoV-1 genome looks like, but it's high level. This is a coronavirus, and so what does it mean? Like what are important factoids you can take away from this figure? Well, number one, this is around a 30,000 base pair or 30,000 character. If we're thinking this as a string, 30,000 character genome. It's small, but complex. It's another thing we can take away from this figure from this previous publication. So we see a complement of non-structural, structural, and accessory proteins across the genome. But really, it's a 30,000 character string that we really want to dive in and deeply understand what's going on at the genome level, at the consensus level, and also at the minor variant or intrahost SNV level. Okay, and then just to segue a little bit, one initial question alluding to my first slide: how does SARS compare to SARS-CoV-2? So next discuss that. So for those that are not familiar with these concepts, I will keep it pretty high level. But when we look at sequence similarity, the similarity between if we were to take that string which represents SARS or SARS-CoV-1 and compare it to SARS-CoV-2 and count all of the differences in similarity. So essentially 100% similarity means every single base pair in the 30,000 base pair genome, when we compare it from SARS-CoV-1 to SARS-CoV-2, is the same. And then 0% would be they're all different. So putting these numbers into perspective, certain non-structural proteins between SARS-CoV-1 and SARS-CoV-2 are identical. And this would be NSP7 and NSP13. So NSP7 is involved in the RNA polymerase complex, and NSP13 is a helicase. And those are 100% identical across their length. So those two, if we're comparing SARS-CoV-2 with SARS-CoV-1, they're identical. And then we get into some more diverse regions. So if we look at other regions, this is now a non-structural protein in an ORF, open reading frame. So we have SARS-CoV-1 and 2 again, and this time it's 86% similarity and 85% similarity. So again, if the length of these, which it isn't, is 100 characters, that means these two differ by about 14 positions across that 100 character string. So and these two are this one's involved in NSP3 is involved in viral replication, and then ORF9b is thought to be involved in type 1 interferon antagonist. Okay, so we have identical, we have clearly not identical, and about 14 or 15% different, and then we have regions that are really, really different. And I say the unknown, where there's this ORF called ORF8 in SARS-CoV-2 that has very little sequence similarity to SARS-CoV-1, not really the same thing. So just to give high-level landscape, when you're comparing these two genomes, you have some identical regions, you have some diverse or more different regions, and then you have very, very different regions. Okay. So next, what I already mentioned these two terms. What are we talking about when I say SNP or SNP and ISNV or intrahost single nucleotide variant? What do I mean by that? Well, I have an example here for you. So essentially, when we go to sequence SARS-CoV-2 from COVID-19 positive patients, we're going to take a nasal swab or other sample, and we will swab that, and then we'll put it on do some library prep and lab prep, and then we'll put it on a sequencer. But as you see in this black box here with the green circles, when we do that, what we're actually doing is sequencing a population of genomes. It's a population of individual virions or cells corresponding to SARS-CoV-2 population inside of that that nasal specimen, nasal swab, or whatever. Okay, so we have all of these different colors which represent different actual individual virions corresponding to SARS-CoV-2. Okay, so but the sequencer will give us all of these black boxes that we call reads. So these are these are sequencing reads, and then so we see CGAT. That's one such sequencing read that will come out, and then genome assembly algorithms will stitch all of those together, and we'll reconstruct a consensus sequence that we list at the top there. That's essentially putting the puzzle together. You can think of the DNA sequencing, the sequencing reads as the puzzle pieces, and then the consensus sequence at the top is kind of the puzzle put together. So when I refer to SNPs, or single nucleotide polymorphisms, I'm referring to changes that occur at this consensus level. If we stack all of the reads up and we make a consensus call, this would be the position inside of that genome or consensus, okay? And then when I talk about intrahost single nucleotide variants, ISNVs, or minor variants, I'm referring to individual changes that we could find inside reads and maybe don't reach the consensus level, but they're present. So I have an example highlighted there. There's two C's on the right that are contained in two reads. And so that would be considered an ISNV. So just consensus level is when we agglomerate all of the information. We can think of it simply as if 50% or more of the individual reads at a position support a given base pair, that will be the consensus, and anything below that will be an ISNV. Okay, so that's an important. I'm going to refer to these terms repeatedly moving forward. And then furthermore, once we now have these consensus genomes across patients, so intrahost, we have the reads, and then once we have this consensus genome, we can now use that consensus to compare SARS-CoV-2 similar, from COVID positive patients across the length of their genome, and these are called, again, single nucleotide polymorphisms, or SNPs. So we see some highlighted regions of what SNPs are, and on the left is a mock phylogenetic tree which relates these genomes based on their evolutionary history. So again, this is primarily to drive home the difference between a SNP and an ISNV. Okay. With that said, we're going to talk a little bit about data now. So now we have ISNVs and SNPs covered and a little bit of the biology. To do any of the analyses I'll present, we need access to data. And fortunately, this has been a global effort of many sequence depositing their sequence data inside of NCBI GenBank. So to date, we have around 5,000 sequences inside of our publicly accessible database at NCBI. And then we have over 6,000 SRI sequences, which would be the read data sets. Okay, so we can, if we were to click on this link, which I won't do, you can go there and anyone could go there and access and download all of this data. Okay, so that's kind of step one. Another great resource and primary resource is GISAID, GISAID, and this here we're going to actually have access to over 32,000 complete SARS-CoV-2 genomes. These are assembled data, as I described in the previous slide. A login is required. You have to go there. You have to create an account. But once you do create an account, you can download these data and you can look at them and compare. There are some restrictions, but essentially the data sets I'm describing are from NCBI or this data set. This is where we're going to get our genome data. And then other sources, along with collaborators on in the COVID-19 International Research Team, there's a compiled open Science Foundation repository where you can access data that I will discuss today. There is a COVID-19 data lake hosted by C3AI with lots of great information. And then, of course, probably one of the most well-known resources is the amazing resource NextStrain, where if you click on the link, they've been just doing an amazing job at keeping up to date tracking of all of this information, pulling the genomic information and showing all this. So there's a lot of data out there for conducting the analysis that I'll describe. Okay. And then finally, I will just show an image of a phylogenetic tree of all of the GISAID genome assemblies contained in that database. I said there were now 32,000. This phylogenetic tree is based off of 7,000 of those genomes. And I'll walk you through this figure. This figure is a little bit complex, but we'll walk through it. So at the inner level, there's a phylogenetic tree that was created based off of the genome alignments. But essentially, we can think of this as we're looking at SARS-CoV-2 genomes, and we're any genomes near each other or evolutionarily related. And so I don't have time, unfortunately, to get into this a full description, but the next layer is going to be the continent from which these genomes originated. So we have this phylogenetic tree, which is looking at the SNPs and primarily the SNPs and how genomes are different or differ and how similar they are. And we build this tree, and then we're looking coloring on the next. So we see purple and black and red. These are all indicating continent of origin. So we can label this with these colors. And then on the outer layers, we actually see what's been called clade defining SNPs. And these are the single nucleotide polymorphisms I previously described that essentially are specific to certain sets of genomes inside of these clades inside of the phylogenetic tree. Okay. So just as a little bit of a teaser, kind of global information, 7,000 genomes, how they all relate by continent and by these SNPs that I referenced previously. Okay. So now the analyses that I'll do moving forward will kind of primarily rely on this for the ISNV and SNP analysis on SARS-CoV-1 samples, SARS-CoV-2 samples. We'll compare these to MERS, another coronavirus, and we'll compare these because we really want to look at how SARS-CoV-2 is different from these other coronaviruses, and specifically look at this intrahost diversity of SARS-CoV-2. So we're getting into part 2 now. Part 2, we're going to cover a high-level summary of intrahost variant landscape. As a reminder, I just recently presented this, but as a reminder, when I say intrahost, remember within a given host, we're going to have all of these green circles, which each contain a copy of a genome for a specific SARS-CoV-2. And we're going to now really look at the differences at the read level, not at the consensus level, but at the read level, and what's going on within a individual, a single 19 patient. Okay. And so what do we mean? How do we analyze that data? So essentially, we take an existing genome assembly for SARS-CoV-2, maybe the first ones from Wuhan, and from Wuhan, the Wuhan reference genome, which I one of my initial slides, and reads and see how they compare to that Wuhan reference. So we're going to use algorithms, bioinformatic algorithms, to actually align them to that reference, and then we're going to look at them and we're going to see all the differences inside of those reads compared to that reference. Okay. So if we dig in a little bit deeper, this is what we call a aligned region, and this is a sequence alignment of the pileup of all those reads to that reference, and we see mostly that everything looks differences in these reads from an individual person. We want to understand what are all these differences caused by and what's going on within person. So highlighting a little bit, we can now see very faintly some different in position 3 read support that are different from that reference, and we see some other positions as well. And again, importantly to clarify, we can map this to a reference, or we can actually assemble out the genome for a given person, and then map all the reads back to that to find these ISNVs. Okay, and then one of the major challenges here, is how do we know this lower frequency, this intrahost SNV is not a sequencing error, because the DNA sequencing technologies can also generate errors when you get one of those reads out. So how do we know that this is not just an error, or if it's actually true variation within a sample? And fortunately, we have some tools available to us that can help tease out error from actual ISNVs. And one of those tools is what we use is low freq, but unfortunately, I don't have time to get into the algorithm, but thankfully we have tools like that out there that can help us with this. Okay, so once we compare all of those reads or map all those reads to that within that single patient genome, we're going to look at what are all the differences, and there's certain differences that we can observe here, and this is what this first plot is, and this is what I'm calling mutational landscape of SNPs or SNPs and ISNVs. Okay, so if we look at how, if you remember all the previous slides I presented, if we look at exactly all of the individual differences we see within a person, but also at the SNP level across genomes or interhost, we see A to T changes, A to C changes, A to G, C to T, C to G, G to T, etc. Okay, on the left. And so this is one of the first observations I wanted to share with everyone was if you look at all of the colors, we have SNP levels as I've described from Houston and New York City. We have interhost variants also from Houston and New York City. And then we have those GISAID genomes, which we've calculated SNPs. So just highlighting a couple of these, we see something like an A to G change, pretty much consistent across all those different types of variants, whether it's intrahost or interhost or SNP level. Another interesting observation, however, is the C to T changes. We see in some of the samples, such as the Houston ISNV and the GISAID SNPs, to be around 30 to 40%. If we look at the Houston and New York City SNPs, we see higher, much higher. And then if we look at the New York City from the Chris Mason lab that was provided the sequence data, that we see that actually the interhost SNVs for C to T changes is actually below 20%. And this is what I mean by landscape. We want to dig in and see what are all the changes within person and across hosts for this virus. And on the right, we see kind of a similar story in the sense that we're looking at these specific changes, but now we're comparing it from SARS-CoV-2 to SARS to MERS. And we see some interesting patterns again. SARS certainly sticks out and certain changes looks a lot different. MERS and SARS-CoV-2 look pretty similar for some of these changes. But once again, we see some interesting patterns in C to T changes where we see SARS-CoV-2 and MERS maybe closer to one another than what SARS is. So this is what I mean by digging into the landscape, and we start to see patterns. Interestingly, there was a recent paper published in Science Advances by Giorgio et al., and they found similar patterns in their data, looking at similar things, C to T changes, but they also found slightly different patterns where they saw a lot of T to C changes, maybe more than what we saw. In a different figure, and when they're looking at the data a different way, where you're looking at SNP level changes, the C to T changes do become dominant. So it's very interesting, and something I wanted to point out for there are recent studies which highlight some of these changes, but this is kind of exploratory analyses based on all the data sets that I described out there. So the next part I would like to do is a comparative analyses of SNPs and ISNVs. And we want to look at them and say, what differences can we observe between them, or are they just the same patterns? So what we have on the screen now is an upset plot capturing overlaps or intersections between SNPs and ISNVs in the SARS-CoV-2 data. And what I mean by overlaps or intersections, it means these intrahost SNVs are occurring at the same positions where SNPs occur in genomes. So if we look at a genome, again, we said it was 30,000 base pairs long. We take position 1,000. We find in some genomes that there's a SNP there. And then some intrahost data, if we're looking at the reads, we find we map them or align them back to that host-specific genome assembly. We find in that position 1,000 there's an ISNV there. Okay. So that's what we're looking at, and we'll get into why we think that's interesting or why I think that's interesting. But as we can see in this upset plot, there's a lot of GISAID SNPs that only exist as SNPs on the far left. There are a lot of those. Same for the New York City ISNVs. But what I find really interesting is when we start looking at intersections in the upset plot, where we see intersection from the New York City ISNVs and the GISAID SNPs. So these are interhost SNVs that occur in the New York City samples and also are found as SNPs in the GISAID genome data that are contributed by the broader community. That's very generously so, and anyone, once you apply for a count, you can access them. Another really interesting observation is when you look at sets of 3 where you can find, for example, in New York City SNP, ISNV, and Gisade SNP. There's 13 of those where they occur. So this is what I mean. I'm not going to go through all the numbers here on this figure, but overall, when you start looking at these intrahost single nucleotide variants, these SNPs in the genomes, you start seeing some really interesting patterns in terms of within person variation and also across the genomic data across multiple individuals. So another way of looking at this is you take all of the SNPs again, and you look across the genome. Remember I said it was a 30,000 base pair genome, and left to right, you're essentially seeing positions. If there's a bar there, a tall blue bar, it's essentially going to indicate positions that are variable with respect to SNPs. So this gives you some sense of across the genome how variable it is. And with respect to SNPs, again, when we're going back to that phylogenetic tree I showed, 7,000 genomes, we're seeing all these SNPs, and you see some really interesting positions that are highly variable. And then the top row is, again, getting back into the non-structural proteins and structural proteins and of specific to SARS-CoV-2. So you can align these variable positions. For example, in NSP12, we see a position around 15,000 base pairs that's highly variable, followed by a couple more. Okay, so we're looking at this global landscape of SNPs across the genome, and we're also doing a deep dive in within the host and looking at what's going on, quote unquote, behind the scenes. Okay, so now if we want to look at simply something that that I'll kind of briefly cover is we want to look at, are these changes synonymous or non-synonymous? Okay, we want to know if these SNPs or intrahost single nucleotide variants result in a synonymous change or a non-synonymous change. And what I mean by that is there's redundancy in the genetic code. So different codons, so different triplets of AGCs and Ts can code different amino acids. Sometimes you can change one of those and still have the same amino acid. Sometimes when you change it, it results in a different one, which will likely that'll induce a non-synonymous change, which could have an effect on the protein structure. Okay, so this is all this plot is showing. We won't get into the specific details here, but essentially we wanted to compare what did we see with respect to New York City, SARS, and MERS. Okay, so it's an interesting pattern where we see that inside of the SARS samples, we saw a very low mean SN value and mean pi sample, which is giving us some sense of the diversity and complexity of the interhost single nucleotide variants within a person, and we see some differences there. So that's all. This really is the main takeaway from here. We see some differences across these different coronaviruses in terms of complexity and diversity. And then if we get into the non-synonymous and synonymous mutation ratios, so actually now dividing this and getting into a ratio, we see that again, there are differences, okay? And some interesting things here are, again, the differences between SARS and the New York City interhost single nucleotide variants and then the MERS sample. So that dotted line at 1, anything above that, this is out of positive selection, and then if you're at 1, you're neutral, and then below it, it's thought of as kind of purifying or negative selection. So essentially, are these changes, what's the result of these changes? So most of these changes, what does this mean? If you're below that line, most of these changes, most of them are going to be synonymous, okay? And then for the ratio. And if you're above the line, you're going to have more hints of more non-synonymous changes with respect to synonymous changes, if that makes sense. Okay, so if you're at the line, there's an equal ratio there. If you're below it, there's more synonymous changes than non-synonymous. And if you're above that line, there's more. So we see some really interesting patterns, okay? And then on the right plot, we're comparing the intrahost single nucleotide variants with the SNP data, and we see also some slight differences there as well. So we're thinking about this in the context of non-synonymous versus synonymous mutation ratios. So this is another really interesting data point for us thinking about the landscape. Okay, and then we're getting into part 4. We're going to think about the potential impact of SNPs and ISNVs on molecular testing, right? So we've seen some patterns now. We've looked at kind of complexing a little bit of the diversity of these ISNVs. But what are there hints of a potential impact on molecular testing? Okay, so high level, just backing up a second, available methods for molecular testing include direct detection approaches such as real-time reverse transcription PCR probes, so qRT-PCR, TaqMan array probes, other probe-based technologies, even CRISPR-Cas-based technologies. We can also use sequencing-based approaches where we do direct, metagenomic, metatranscriptomic sequencing, PCR amplicon sequencing, and targeted amplicon sequencing. So these are kind of our available tools inside of our toolbox for testing for the presence of COVID-19 on the molecular side. And there's other techniques as well, but we'll focus on these. And what are pros and cons for direct versus sequencing? So direct detection approaches tend to be faster, can be, and not always, but overall tend to be faster. You can go from sample to answer sometimes in a few minutes, or 30 minutes, or 90 minutes, or a few hours. Can be multiplexed for multiple samples or targets. And then sequencing-based approaches provide the full genome. You're going to get access to the full genome instead of targeted regions of the SARS-CoV-2 virus. And we can use that to detect co-infections. Typically, it is slower and more expensive. And importantly, typically the sequencing-based approaches require additional bioinformatic analyses. So those are kind of our options here. So the direct detection approaches have been pretty commonly used for COVID-19. If anyone's seen the nasal swabs or nasopharyngeal swabs, you get swabbed, and then you get that swab will get analyzed typically through one of these direct detection approaches for a fast and usually reliable result. But why this is important in the context of what we previously discussed is, again, as it's kind of targeted, you're looking at specific regions in the SARS-CoV-2 genome. Again, 30,000 base pair genome. We're going to target a few regions, and we say if we see these regions present, we're going to call that as a positive. It's more complicated than that, but given the limitations of the time I have for the presentation, we'll just leave it at that. But we're going to target maybe 3 to 4 regions across the genome with these specific 20 base pair to 30 base pair sequences, so that we can say if we see these as present, we're going to say, okay, SARS-CoV-2, this is a COVID-19 positive patient. And if not, it's going to be a negative. Okay, so we have listed in the left figure and the right figure. The left figure are the intrahost single nucleotide variants, and on the right are the SNPs. And each row is actually one primer probe and available direct testing approaches based on what I just previously described. So what we did is we took all of those and then we overlaid the ISNVs and SNPs on top of them. And so the landscape of that we previously observed, we see that many of the primer probes actually do have presence of SNPs and ISNVs in them, okay? And we haven't done the validation of actually demonstrating this is going to make it so that, result in a false negative test, but this could reduce the efficacy or could result in a false negative for some of these commonly used tests. So this is something good to keep in mind in terms of the implication of the SNPs and ISNVs we previously discussed. And to kind of solidify this point, a recent paper that just came out at the end of May described exactly this: the presence of mismatches between diagnostic PCR assays and coronavirus SARS-CoV-2 genome. So I listed that paper. It's a really nice read on kind of going in there and understanding the effects of the mismatches between the genome and what I just described. Okay, but for the interest of time, I will have to move on. And then I'll end with the potential utility of ISNVs for COVID-19 transmission analyses, and I'll describe exactly next what I mean by that. Okay, so why do we care about transmission? Well, essentially, when we spread SARS-CoV-2, we don't just spread, just this consensus sequence. It's possible we're spreading multiple copies of this genome via the cells or the virions. So in this figure, we have a donor. We have the virus gets in, it replicates, and then let's say we cough or sneeze or however we transmit it. The recipient may get one single copy or more, and that's what we'll talk about. But essentially, these individual kind of cells or virions that we can look at through the read data, as I mentioned, could be helpful for saying this recipient actually received the virus from said donor. Okay, and that's kind of number one. Number one is kind of the first thing to think about. And then number two, though, there's this concept of transmission bottleneck. It's the number of viral particles initiating each new infection. Okay, so if the donor has a lot of virions, so the role of the bottleneck size, so we have a large population, this bottleneck size kind of has a different effect when the viral population is small. So think about it, we have a diverse population. We want to understand how many possibly of these virions would transmit to a recipient. Okay, so moving on to the next slide, just looking at this slightly differently and similar to what we've talked about previously, putting it all together, we have some donor with some copies of the virion. This was our original figure. We sequence those, as I said before. We identify some of these have some ISNVs, right? We can look at the frequencies of those ISNVs, and then we can look at the recipient, maybe a putative transmission pair. Maybe we know it's a family member that is CoV-ID positive now, that has a few copies. We can sequence that, the recipient as well. We can look at the reads. We can look at the ISNVs, and we can compare. We can say, does this profile look like it's similar, or does it look dissimilar? And there's many factors which would contribute to that. But in the interest of time, that's kind of what I wanted to bring up. So this is what we next did. So in that same data set that I described, we looked at shared intrahost single nucleotide variants, again, going back to the same position across pairs of individuals in this New York City study provided by the Chris Mason lab. So we looked at all of this, and we found some really interesting pairs. While the top left figure A, most pairs didn't share anything, but there were a few pairs inside of this data set of 140 samples that we saw shared interhost single nucleotide variants. So on the top right, we see that the number of samples that contain a given ISNV varies. There are some that are very prolific that occur in many samples, so maybe they're not informative. We see an ISNV, and there's some technical reasons why these could be artifacts, et cetera. We see them occur in a lot of genomes, and then we see on the right hand of the figure panel B that there are some that occur in a very fewer genomes, okay, maybe less than 10. Then the last thing we can do is using some modeling algorithms. We can look at on the left this bar plot that's kind of a dark gray, and compare it to this dark green. The dark gray is let's say it's a putative recipient. We don't know, but let's say for the sake of the presentation, and the green is potential the donor or vice versa, and we can look at the common ISNVs across those two patients, and we look at the frequency at which they occur, and we found some really interesting patterns. We see some positions for some of these ISNVs are at very similar frequencies from putative donor to recipient for both C1 and C2 in this figure. Okay, and all I want to say with that is this hints at potential utility of these ISNVs for tracking transmission of SARS-CoV-2 across patients. And of course, there are some recent studies on this that I won't have time to describe. All right, so with that, I will get into my conclusion. I will just say, start by saying diverse intrahost variant landscape in SARS-CoV-2, including ISNVs, SNPs, and other variants. And this varies from genomic data, as I described, from New York City samples to Houston samples that were analyzed for this study. Multiple intrahost ISNVs and SNPs were found within commonly used probe and primers for molecular PCR detection. This is something that but just to highlight, should be taken into account for primer and probe design to ensure effective detection and limit false negatives. And then the last point is multiple ISNVs were shared between hosts, and some at high allele frequency. This suggests the wide bottleneck and potential also gives us some hints at their potential utility for tracking transmission. Okay, and with that, I would like to acknowledge many people. There's many people that contributed to these analyses. I just was fortunate to be able to present this to you all. But first, I'll start with my lab. My postdoc in my lab, Leo Elworth, major role in all of these analysis, and PhD student Nicolas Sapoval, who led most of these analyses. Maria Wang, who contributed heavily to the transmission analyses. Yunxi Liu, Tracy Albin, and Yulei Fu. All major contributions to the work, and hats off to them for doing all the analyses. And then I want to say, also highlight Dr. Fritz Sidlacik's efforts, and as he's kind of the co-senior author on the study, leading the charge on the structural variant analysis, which I didn't have time to cover today, and his postdoc Medhat Mahmoud. And then others. I won't read through all the names, but there's everyone on the slide deserves acknowledgments, and then special thanks also to the Mason Lab, specifically Christopher Mason, for providing a lot of the samples and the data for the samples that were presented today. And with that, I will go off on a slight tangent. I would like to highlight the COVID-19 International Research Team. It's my great honor and privilege to do this and share this with you today. I am fortunate to be a co-lead of this international research team alongside Afshin Beheshti. This originated from a NASA GeneLab multi-omics analysis working group that Afshin leads. I was a member of that, and we proposed working together on COVID-19 research back in March. So since then, thanks to all of the members, this is just that have joined and contributed all of their nights and weekends and their tireless efforts to it. This has grown immensely, and it's very excited to share this with everyone today. Please do check it out. But yeah, many members with diverse expertise all working together in an open science fashion. And so it's been very excited to be, I've been very excited and privileged to be involved with this. We have a website. It's www.cov-irt.org. This is the landing page for the website. Please do check it out. I can provide the links later. And then importantly, we are hosting our second symposium, Friday, July 17th at 10 a.m. Central Time. And this is a save the date, more details to follow. We're finalizing the speaker agenda now. It's coming together really, really well. And so I'm excited to soon share that with everyone. But if you're interested, please do save that date, and more details to follow. And then just give you an idea a little bit more about Covert. We are, I said, international research team. I'm very excited to say we're in multiple countries across the globe, across the U.S. and across the globe, and we're actively recruiting new members in some of the countries where we don't have representation. But again, this has been just all thanks to all of the members that join and contribute all of their time and effort and work together on this very important problem. And just to acknowledge all of the members. This is not all, but this is the latest up-to-date one that I had. It's missing a few members, but I believe we're 120 members and counting, potentially more at this point, several more. But we have representation across the U.S., across the globe, many different institutions, and yeah, just really amazing, strong team, and again, very fortunate to work with everyone. So with that, I will conclude my presentation, and happy to take any questions you might have.
Thank you so much, Dr. Treangen, for that informative presentation and clearly important research. We will now start the Q&A portion of this webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's take a look at the incoming questions that are already here from our audience members. And thank you again, audience, for these great questions. Dr. Treangen, why are the SNPs insufficient for tracking transmission of COVID-19?
Yeah, that's a great question, and something that could be a presentation on itself, but I had to kind of maybe go a little fast there. But essentially, when we look at the genomes of two SARS-CoV-2 viruses that were transmitted from person A to person B, it could be that they are identical at the consensus or SNP level. So we look at we sequence the genomes that I described. We assemble those genomes for the specific, patient A and patient B, and then we compare them as I described. And we look at across the 30,000 base pair genome. If this is a direct transmission event, it's possible that these genomes are identical or nearly identical. So the reason they're insufficient is that there could just be no SNPs that they're essentially identical. So we can say, okay, well, they're identical, and this could be the same for a cluster of recent genomes that are all identical. So we could say, okay, well, they're all within a pool of very similar genomes, maybe differ by 0, 1, or 2 SNPs. But we can think of ISNVs as extra kind of firepower, or quote unquote, to actually look into and say, okay, well, if we have multiple genomes from multiple patients, not just A and B, but now we have patient A, B, C, and D, and E, all identical. But now we find a pair inside of those 5 that also share interhost single nucleotide variants. This gives us extra confidence, additional confidence to say this likely represents a transmission event. So it's not that they're always insufficient, but sometimes they may be insufficient because of, for certain transmission scenarios, it just could be that it's part of a larger indirect transmission route. But if we really want to use this information to say this looks like it was a direct transmission, we could maybe get confused by other genomes that look just as similar without looking at the intrahost single nucleotide variants.
Thank you. And regarding the SNPs, can SNPs cause the non-detection of positive samples by RT-PCR?
Yeah, this really varies. That's a great question by platform. Some platforms are more sensitive to SNPs or SNPs that could occur in the genome, and some are more robust. But some platforms, if you have 1 or 2 or a handful, this could really interfere with the detection, or even just 1. And other platforms are more robust to this. And again, these aren't things I had time to really fully address, but based on the specific strategy in the platform for the RT-PCR-based detection, it could be that a handful of SNP changes does interfere with a positive detection call. So it's something we really want to think about, and it's thought about when these are being designed. But something I didn't get a chance to mention is, even if we design this with our best algorithms and methods out there, and there's a lot of great methods, I won't be able to list them all out. Sometimes we just don't have all of the up-to-date information. Again, so this really started ramping up, let's say back in March. Let's say we did our design of these primer probes for detection back in March. Well, now 3 months have passed, and it could be that now as part of the we see additional viral genomes that have SNPs in the positions where we previously thought were highly conserved or identical across. So it's not that it's just the kind of the hard part of this whole problem as merging situation as additional data arrives. Both SNP and ISNV data is really crucial for understanding what's going on. And so as additional data arrives, and this is another figure I wasn't able to show, but let's say we go back to March and we have a handful, maybe tens of genomes, right? And then we get into hundreds, and then we get to thousands. And now I mentioned in one slide we have over 30,000 genomes, and this is growing so rapidly that all of this is additional information for us to look at and say, how do we design effective tests that are robust to changes? And we have more information now. We can do that, and we're getting a better sense of the mutational landscape. So yes, it can. Really varies by platform. So I'm not going to get in. Some will work better than others, but it really gets into how these are designed. But it's something certainly to think about, and it's something as more data emerges, it may update our understanding of what these conserved regions are. So as there are more changes introduced, so over time.
And Dr. Treangen, what is the difference between ISNV and SNP?
Yeah. So this is something that is, for those that haven't thought really carefully about this for a long time, and even sometimes it's confusing for me when I'm describing this. But it's essentially, again, when you're thinking about for a given person, when you sequence a given person a swab, let's say we have a nasal swab, and we put it on a sequencer. We have a collection of reads. And then we're gonna create a consensus genome assembly, And then we're going to align all those reads back to it. And so any ISNV in that scenario is going to be anything that occurs at less than 50%. So let's say for a given position, we align or map or 10 reads to that position. And if we see a T that occurs in 3 of those 10, that would be an ISNV, meaning that it doesn't reach this consensus level, but it's present in the population. And we don't think it's an error. We think it occurs too frequently to be an error, but it occurs not frequently enough to be a consensus level kind of SNV or a SNP. Okay, and then SNPs are when we think about when we're just dealing with genomic data from GISAID. So we have these 33,000 genomes, and we're comparing the genomic information. Forget about the read information from the DNA. So we already have the genome assemblies, and we're comparing all the genomic information, the genomes, the assembled genomes, which are assembled through the consensus of all of these, all of the data I mentioned. Those would be SNPs. So we're thinking about consensus level or greater differences. So, and we can think of these as interhost also. Another way of thinking of this is intrahost versus interhost differences. It's another kind of maybe a little bit more cleaner way of thinking about this is looking at changes in SARS-CoV-2 within a given person that you might not observe in other individuals or patients. And then SNPs are more interhost, things that you observe across patients. So, and they can intersect, which I described in the presentation, but that's kind of the main difference. Again, when we think about we're COVID positive. We have the virus, and it replicates, creates many copies, and then there's this population of virions, and each one of those might contain its own slightly different version and differences. But then so that's one way to look into this problem, and then another way is actually go for all of the assembled. The consensus sequences or the genomes that have been assembled from each of these patients and compare them to each other. And then we see kind of interperson or cross-host differences that might be of interest. So I think one of the main points of the presentation is really understanding that there's kind of two sides of this to think about and consider for these viruses. And we really want to kind of combine that information, look at the consensus level information, also the interhost with the intrahost. So I think that's kind of the one of the main points of this the presentation. But yeah, that's a great question.
Thank you. We have time for a couple more questions. And again, thank you to our audience. Any questions not answered today will be answered via email. Will the variations in the gene sequence of the spike protein, could it hamper the vaccine development?
Yeah, I mean, I'm a computer scientist, and I don't want to get too into any. At this stage, I'm thinking about this as an exploratory. That's why I use the words landscape and looking at this. Certainly, when we project this out and think about it more carefully, which I'm not ready to really kind of get into this because I want to look at more and I want to look at a lot of the other scientific results and think about this more. But certainly, there's certain mutational patterns that may cause more than be challenging in different ways than what I just described. The two methods, the two kind of primary kind of are what's the impact on kind of molecular diagnostics, and what's the impact on maybe utility for transmission. So certainly that's one of the main goals of where we're at currently. The team that I acknowledged and even Covert broadly is looking into all of these changes and then trying to overlay this with other potential kind of negative impacts or consequences, and thinking about vaccines, but again, there's nothing really kind of for me to add there other than it's an interesting question, but more broadly thinking about all of this in the context of the virus and of the host, and also the part that I didn't talk about today, but there's also a microbiome component. So just putting that all together, and again, that's just wanted to stress why I think it's such a critical thing to have a team of diverse expertise. So when I'm looking into these patterns, intrahost and interhost diversity, I could then connect up with an expert in the area of vaccine development and say, how would these changes impact anything, or would this help in the design? So I haven't, that's a great question, but I think more follow-up research is needed to really precisely answer that and then look into it more carefully.
Thank you so much. Our final question, what sequencing platform was used to obtain the consensus and read sequences?
Yep, that's a great question. I kind of skipped over that a little bit. So for the New York City and Houston samples, that was an Illumina NovaSeq. Okay, short read sequences, low error, shorter sequences. I think 100 to 150 base pairs in length. And then some of the J8 samples, it's a mix. I don't have the statistics memorized, but it's there's a lot of Oxford nanopore data, and then there's a lot of Illumina sequencing data there. I think those are the two dominant ones. And then inside of that, there are some variance and variation, but overall, it's to answer your question, for the analysis that I presented today, we primarily had J8 genomes that were kind of constructed from Illumina sequencing platforms in Oxford nanopore, and then for all the intrahost diversity, or most of the intrahost diversity analysis I present today, was Illumina NovaSeq 6000. So that's what was used.
Thank you so much for your presentation today, Dr. Treangen. Would you like to provide any closing remarks for our audience before we leave?
Yeah, no, just thanks again to Labroots for inviting me to give this presentation, and thanks to you, Susie, for hosting and moderating. I really appreciate it. And feel free, because I'm happy to field any follow-up questions, and would really encourage everyone to go check out the Covert website, www.cov-irt.org, if you're interested in getting involved. And it's been a pleasure to present today on this platform.
And thank you again, sir, for your time today and for your important research. And before we go, I want to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted on the on-demand period will be addressed by the speaker via the contact information you provided at registration. And this webcast can be viewed on demand through the end of the year 2020. Labroots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, thank you, be safe, take care, bye-bye.